All right, pretty cool. All right, let's get started on another Climate, People, Environment Program seminar. We have a intimate audience today, but I'm sure we'll be more popping in a bit. But it's my pleasure to introduce Nicole Ward, who is with the Minnesota Department of Natural Resources. And I'm going to read from her bio here, so you get a little bit of flavor of work she's up to. But Nicole's work's been driven by science-informed management, as she says, since middle school. That's a pretty long time knowing that that's what you're going to do. So I'm very in awe of that. But she's going to share some of that with us today, including her work of connecting academic research with managers and stakeholders. I think something that many of us aspire to do. So I'm excited to learn a little bit more about how that actually works in practice. She got her undergraduate degree here in UW-Madison, and she currently is in Madison for a little bit longer before heading over to Minnesota. She's been working remotely, so she's trying to soak up as much Madison culture as she can. But now is with the Minnesota Department of Natural Resources, and she worked there previously as an aquatic biologist before going on to the University of Idaho Water Resources Program, where her master's research also had stakeholder component with farmers. She then went over to work with Professor Kaylin Carey over at Virginia Tech, which is where she did her PhD, working with a nonprofit lake association. And then she came back to Madison and was on a postdoc at Wisconsin Sea Grant. That is, I think, when I had invited her and when I think a few limnologists suggested that she gives great talks. But very soon after her arrival, she got recruited by the Minnesota DNR, which is where she is now, soon to be stationed in Lake City as field station supervisor for the Upper Mississippi River Restoration Program. So today we're going to hear a little bit about lake modeling and socio-ecological systems. This is projects that Dr. Ward has worked on in a number of different ways, and she has published a number of papers on these topics. And so definitely encourage you to reach out afterwards if you have more questions. And with that, I'll turn it over to Nicole. And if you want to go ahead and start sharing your screen, and I will go off video.
Awesome. Thank you so much for that intro. Let me see, get my right screen here. And if anybody feels so inclined to keep their videos on, I love to actually just know that there's humans on the other side. But if you guys want to keep your videos off, that's also totally fine. But it's yeah, at least halfway through the talk that will help me, keep energized through it. Okay, so thanks so much for joining remotely. I had been really looking forward to doing this in person, but I appreciate kind of the last minute change as I am feeling a bit sick and thought it probably better to keep my germs away from all of you. So yeah, thanks for taking your time this Tuesday afternoon to hear about my work. Okay, so today I am and I just want to make sure you guys can see you see the right screen? Is that the funding and support?
Your presentation.
Perfect. Okay. So today I'm largely going to be talking about my dissertation research, which was supported by the top line of things here: NSF, Virginia Tech College of Science, and the Lake Sunapee Protective Association. And then towards the end, I'm going to share some kind of new research directions that I'm taking in this new role at the Minnesota DNR. I've only been in this position for about 3 months, so it's all very new and kind of in transition. Okay, so often environmental management and science are considered as these kind of two separate things, where in kind of good situations, science is informing the management. And I would say in what I would consider really great situations, the management is also informing the science. But for me, this kind of distinction between science and management has always been a little bit more blurry because I learned how to conduct science within a management context. This goes back to when I was in 7th grade. This is a newspaper clipping from 2001. And the lake that I lived on in Minnesota was invaded by zebra mussels. And I conducted a series of science fair experiments kind of tackling this ever-changing management need from 7th grade through 12th grade. And so because of this kind of formative childhood experience in conducting science for management, I've kind of always thought more in terms of this integrated science and management perspective, where managers are learning from the scientists, but also really importantly, scientists are learning from the managers. So this is where kind of everybody involved in cross-sector partnerships, so say including state agencies, academic institutions, and even private landowners, everybody comes to the table and really values the perspectives and knowledge that all of the other parties are bringing to the table. Okay, so into some of my specific research, I have been focused on freshwater quality, and I really love researching freshwater quality because water is this very kind of unifying shared resource for people. And for me personally, when I think of my connection to water, I think of my late grandfather, who, when he was just a boy, he helped my great grandfather build our family cabin. And this is on a lake in northern Minnesota. This cabin remains kind of largely unupdated to this day, although we did add a flushing toilet at one point in time. But overall, kind of words can't really adequately express my connection to this cabin and to this lake where we have wild rice lining the shores and loons are kind of constantly calling across the bay. I absolutely love it. And what I love most about when I think about my connection to water and lakes is that my story isn't unique, and it's only one small example of the ways in which someone may be connected to water. So kind of all people throughout all of human history are inherently connected to freshwater systems. We can think, back to the ancient water aqueducts of the Roman Empire, irrigation systems that are over 2,000 years old in China, to the recreational, cultural, and subsistence fishing across the world today, and that have been happening for thousands of years. And importantly, since people are so dependent on freshwater, we're also very vulnerable to changes in freshwater systems. So for example, we have increased flooding due to climate change and drinking water concerns such as toxic algal blooms in Lake Erie, which left hundreds of thousands of people without access to clean drinking water for days. These complex issues in freshwater systems, where people both depend on water and are therefore vulnerable to changes in it, require interdisciplinary approaches, or more specifically, kind of considering both natural science and social science. To kind of conceptualize this, let's consider that we can represent the natural or biophysical system as connections between terrestrial and aquatic processes. And then at any kind of specific point in time, we can consider the state of an ecosystem. So here we could say that there are 19 fish present in this stretch of stream, and there's a high concentration of cyanobacteria in the water. Then when people or society assign some value, whether it's monetary or not, to these ecosystem states, we then think about them as ecosystem services, or that kind of value, benefit, or sometimes harm to people or society. So here we could say that the salmon are a key cultural identity for many indigenous groups, particularly in the Pacific Northwest of the United States. And then the cyanobacterial bloom is limiting access to drinking water. So then these ecosystem services go into economic and socio-cultural processes, where society responds to those changes in ecosystem services through the creation of institutions, the formation of new preferences, and all of that contributes to individual human decision-making and then resulting human actions. Those actions then impact the biophysical system, kind of closing this bidirectional feedback loop. And there's a lot of different ways that we can kind of visualize this. And so I think key to point out, even though I'm representing these as two separate things, they really are integrated, and all of those connections between them are happening kind of constantly and simultaneously, and everything is feeding together. I think because I'm a modeler, I like this kind of box and arrow diagram, but conceptualize it. And yeah, think of it in that kind of integrated, constantly changing kind of state. Okay, yeah, I think using this as the framework for how to think about how we can connect natural and human system processes, specifically to manage freshwater ecosystem change. Now, complex change in coupled human natural systems arises from this interaction of what we call fast and slow variables. So fast variables change hourly to yearly. So for example, stream flow and water color in this headwater stream. Here at 5 p.m., the water is kind of trickling over the rock and the water is clear. A few hours later at, say at 9 a.m. the next morning, that stream is rushing over the rock and the water has turned this kind of brown color. By 6 p.m., it's still flowing pretty good, but now it's starting to turn clear. So this water flow and color of the water are fast variables because they're changing hourly. In contrast, slow variables change over the course of decades. So one type of slow variable that we think of in lake systems is lake trophic state, which is a classification system that helps identify the overall conditions in a lake. So for example, these two lakes look very different. In lakes that generally from year to year have clear water and low algae are called oligotrophic lakes. And then lakes that generally from year to year have murky water, frequent algal blooms are called eutrophic lakes. Overall, lake trophic states generally remain constant for years, decades, even centuries. And oligotrophic lakes may transition to eutrophic lakes through a process called eutrophication. This is a type of ecosystem regime shift, and these occur when an ecosystem goes from kind of one common state to another. So these ecosystem regime shifts in general, we could think of as slow processes. And if we think of kind of lake trophic state as the slow variable, which can then be modified by that slow process of eutrophication. That process is driven by changes in that fast variable, where we have this kind of accumulation of nitrogen and phosphorus in the lake over time from nitrogen and phosphorus load, which is the amount of NNP delivered to the lake. Those fast variables of NNP are constantly accumulating, and as they do, we might not observe that change in the slow variable until those kind of accumulating fast variables have reached some sort of threshold. And at that point, we might see this relatively abrupt shift in that lake trophic state. So this would be where a lake would flip, quote unquote, flip from oligotrophic to eutrophic. Okay, so because of the inherent complexities in human natural systems, and especially with this interaction of fast and slow variables, using computer simulation models helps us kind of unravel and examine those underlying processes and enables us to look at long-term trajectories of change. I'm specifically interested in this, doing this to improve water resources management. And by that, I just mean kind of informing any decision that will affect water quality. This could range from regional watershed management plans and policies to individual private property decisions. So to do this in my dissertation research, I focused on Lake Sunapee in New Hampshire. This lake has a really long history of interactions with people. It's located on indigenous Abenaki land, and the colonial name Lake Sunapee comes from the Abenaki word Seninabe, which means rock or mountain water. Lake Sunapee is this oligotrophic lake with low nitrogen, phosphorus, and algae, and high water quality. The lake is a really important drinking water source and provides popular recreational amenities. It is kind of increasingly threatened by climate change and ongoing land use change. So my primary aim was to help inform how to maintain this oligotrophic lake into the future. But at the time that I was doing this work, I was working as a graduate student in an academic institution, and I was kind of left with this question of how does an ecologist and a graduate student ecologist contribute to improved water resources management? Making that connection between academic research and on-the-ground management is kind of full of difficulties at all the stages. An increasingly popular approach is to work directly with local stakeholders in a process called knowledge co-production. And co-production should be work that is context-based, meaning it's focused on a specific issue or a place, pluralistic, meaning it includes local knowledge, goal-oriented, so making sure there are shared goals between the researchers and the stakeholders, and then it should be an interactive process, so having repeated interactions throughout the research process, and importantly, including interactions before the research questions themselves are even developed. So I adopted this kind of co-production style of research for my dissertation, and I was lucky to enter into a lab where there was already a relationship between my advisor and this nonprofit. And I think without that foundation, I would not have been able to kind of dive into this this type of co-production work kind of from the beginning. Okay, so I'm going to kind of describe the process that I went through as this linear, clear process. but I assure you, kind of in the beginning, I did not see the road ahead of me. I learned kind of a lot through the process. So the path that I took, I'll kind of go through each of these in a little bit more detail, but first was just to engage in partnerships, kind of like I already described, contextualize that local system within bigger picture water management issues, conduct local system assessments, and then finally work on identifying leverage points for change. So first, as far as engaging in partnerships, I worked with the Lake Sunapee Protective Association, as I already mentioned. I'll refer to them as the LSPA. This is a nonprofit in the watershed in New Hampshire that's over 120 years old. It has been that this nonprofit has been focused on maintaining the environmental health of the lake and the surrounding watershed over the course of their time. Partnering with the LSPA kind of worked towards these four aspects of co-production of knowledge. Being context-based, it's focused on Lake Sunapee, pluralistic. I learned a lot from the LSPA and their members as kind of local knowledge. Goal-oriented, working with them. I worked with them and made sure that I was kind of focusing my research in a way that would help them achieve their goals. And then interactive, I met with them regularly, including before my research questions were even developed. Then moving on to this contextualization, this was focused on contextualizing the research within kind of broader coupled human natural system work to give context, to give kind of regional and even perhaps global context to how we were addressing issues within this kind of specific watershed. Then conducting local system assessments. This was both examining how long-term, temporal, and also spatial patterns, and specifically looking at kind of linking fast and slow variables to understand trajectories of change and how this system may be approaching a regime shift. And this was, focused again within the Lake Sunapee region, conducted these assessments with frequent stakeholder interactions, focusing on those shared goals, and then incorporating local knowledge. I would meet with them kind of when I had preliminary data analysis, get feedback on how they were reacting to that data to help my interpretation, as an example of incorporating that local knowledge into the science. Then finally, that leverage point for change to just better reach the management goals of the Lake Sunapee Association. And this could be identifying new management techniques to initiate kind of a specific change in the watershed. And again, this work, which I'll go into detail on all of these, was building on those four kind of main components of co-production. Okay, now as I go through the details of each of these studies, you're going to see this computer icon to represent the types of models that I used. These diagrams, when I'm talking about kind of which components of that human natural system I'm considering, and then this icon when I relate those findings to improving water resources management. Okay, so first, just super brief highlights of this big picture contextualization. This was a literature review of coupled modeling in human natural freshwater systems. This photo is actually taken on the terrace in Mendota. I was on a project with researchers at the Center for Limnology, so we actually had a whole project meeting here in Madison during my PhD, which was fun. Okay. In this study, we used a keyword search to find 601 abstracts, which we then screened to identify 26 papers that met our criteria of being freshwater research that examined this full human natural system feedback loop, and research combining the use of at least one human system model and one process-based natural system model. We found that integrating fast and slow processes is essential for simulating long-term trajectories of change in human freshwater systems, yet those slow processes are rarely represented in those modeling approaches in the literature. And this has really important management implications because if managers want to make those long-term goals, the integration of fast and slow variables is really required to anticipate those long-term trajectories of change. Because we found that those slow processes were underrepresented, we aimed to better understand long-term change in Lake Sunapee. And Through interactions with the LSPA, there was a clear need to better understand the long-term effects of specifically of climate and land use change in the Sunapee watershed, and really how those may be interacting or kind of feeding off of each other in the changes that they're seeing in the lake. So this long-term study did a long-term modeling of climate and land use effects on water quality, and we focused on these blue components of the human natural system feedback loop. So mainly on the aquatic processes and then the resulting states of the ecosystem. We used a process-based hydrodynamic ecosystem model called GLM-AED. The main processes that this model simulates are kind of shown in this diagram. Basically, it's a comprehensive lake ecosystem model that simulates the physical, chemical, and biological processes in the lake. And to use a model like this, we need to do a calibration phase where we first gain confidence that we're kind of accurately representing local ecosystem processes. And here, this line behind the points is the model simulated water temperature on the top panel, and then dissolved oxygen on the bottom panel. And this is just showing from 2005 to 2010. The lighter shaded circles are the manual measurements. So this is kind of where a person went out, took manual measurements on a boat. And then the darker circles are data from our high frequency sensors, which we have deployed in the lake since 2007. So that's why you only see those showing up on the graph starting in 2007. We kind of look at this comparison of the simulated and observed data, adjust those parameters until we're confident that we're accurately representing the ecosystem variables to the purposes of our study. Then with this calibrated model, we conduct 31-year scenario testing to determine the effects of climate and land use on water quality. So this is a close-up of those major processes that are represented in the model. And we tested increasing air temperature by 2 degrees Celsius, which is in addition to the already observed 1.4 degrees Celsius in this particular location since 1970. For land use effects, we tested doubling stream inflow nutrient concentrations. So here N is the nitrogen, P is phosphorus, and those are limiting nutrients for phytoplankton growth. And this doubling of concentrations we picked because it's within the expected changes due to development projections within the watershed. Okay, so we're using these kind of 2 single change scenarios to isolate the effects of that air temperature and then separately the nutrient load. And this ecosystem model kind of spits out a lot of different variables. So we needed to decide what response variable we were interested in looking at. And because we want to look at how climate and land use are contributing to this slow eutrophication process in Lake Sunapee, we know that Lake Sunapee is oligotrophic, but it's in danger of transitioning to this lower water quality state. And a key indicator of that regime shift change in lakes is phytoplankton abundance. And an easy way to estimate phytoplankton abundance is to measure chlorophyll A concentration, which is essentially the greenness of the water. So you could see in that kind of eutrophic example photo, that water is very, very green with phytoplankton, whereas that oligotrophic photo has very low greenness or a very low chlorophyll concentration. Okay, so we pick chlorophyll A as our response variable, and then we run this model for the 31-year simulation from 1985 to 2015. We look at chlorophyll A concentration through time and see this sort of response. Concentrations go up each summer, they go down each winter, as you expect in this type of lake. But to then figure out how the increased air temperature and nutrient concentrations are affecting that phytoplankton response and that unique response in each year, we conducted these scenario tests where we did 4 different scenarios, first having our reference, just like in this graph, then we run the model. We run the model to simulate conditions over the previous decades to the best of our knowledge. We then add this kind of 2 degree scenario, again running it from the same time period, just simply adding 2 degrees to the air temperature driver variable. Of course, this is a simplified not even a climate scenario, it's just a temperature scenario because we know that temperature wouldn't change alone, but we're trying to isolate these kind of processes connecting those driver variables to specific responses in the lake. Then we run the same scenario with 2 times nitrogen and phosphorus, and then our combined plus 2 degrees C and 2 times nitrogen and phosphorus. Okay, so now when we run these, what is interesting is that the effect of, oops, sorry, am I getting lost? Okay, what's interesting is kind of as we run these scenarios, the effect of this 2 degrees Celsius and the 2 times nutrient concentrations in how it relates to chlorophyll concentrations is different whether we look at maximum versus median chlorophyll. And to kind of run you through what we saw, first, I'm going to just show you the maximum response. So this is the 31-year distribution plotted in a box plot. So we're kind of taking each of these annual years and stacking them on top of each other, where we have one dot representing 1 year of maximum chlorophyll over the course of the 31 years. Okay, so here we can see that the maximum annual chlorophyll is around 1.5 to 2 milligrams or micrograms per liter. This is low concentrations. This is a very oligotrophic lake. This is way lower than what you would see in Lake Mendota. Then in the plus 2 degrees scenario, maximum chlorophyll is not significantly different from the reference scenario, but annual median does increase significantly in this temperature scenario. And then both max and median increase with the 2 times nutrient scenario. And then the combined effect of plus 2C and 2 times the nutrients is significantly higher than the nutrients alone. So here, this really big response due to the nutrient increases is not surprising because it's this nutrient limited oligotrophic lake. But what was surprising was this greater change in median versus maximum as a result of air temperature. This indicates that with rising air temperatures alone, we could expect to see that average water quality will worsen in Lake Sunapee, whereas changes in maximum chlorophyll may be more tightly connected to changes in nutrient load. And this kind of fits within established concepts in the literature where average conditions in the lake are more representative of trophic state, or that slow variable, and more extreme conditions in the lake are more representative of those short-term kind of immediate conditions in the lake. Okay, so in summary, this long-term study used this process-based model to examine the human impacts on aquatic processes and the resulting ecosystem states, finding that maximum chlorophyll is more related to external nutrient load than median, and the management implications being that monitoring annual maximum might provide a better assessment of that nutrient load effects, and if managers are wanting to manage to put in efforts to manage that nutrient load, monitoring maximum might be a better kind of response variable to use. And usually these monitoring systems are better set up to track changes in average water quality, so it might be a kind of mismatch between what managers are trying to see in the system versus kind of what they're able to monitor. And then also average water quality is expected to worsen with rising air temperatures. So incorporating that into long-range planning will be really key for this watershed. And then in this nutrient-limited lake, kind of even further reducing nutrient load might help lessen the effects of that rising air temperatures, because the phytoplankton may not be able to kind of make use of those better temperature conditions if there are even less nutrients available for growth. So from that temporal study, we know that mitigating nutrient load is really crucial for maintaining this lake into the future, particularly when those effects are combined with climate change. And then moving into this spatial study, we want to examine any spatial patterns in this eutrophication process. And we specifically focus on cross-ecosystem connections that might be linking the terrestrial and aquatic systems and indicators of lake ecosystem status. So here, we're interested in if there are specific locations in the watershed that are contributing most to that eutrophication process. This lake has many different inflow streams, each of which contribute nutrients to the lake. And a helpful concept to identify how streams are contributing to eutrophication may be what we call freshwater metabolism. This is the balance of respiration and gross primary production, or photosynthesis. This is a sensitive integrated metric of overall ecosystem function because it includes the respiration of all of the biota in the lake, from the microbes to the fish, and primary production from all of the photosynthetic organisms in the lake. Okay, so respiration, let's see. It's 4:30. This is perfect. Okay, so to highlight respiration, I want you all to just take a breath with me. We're going to take a deep breath all together. Ready? In and out. Okay, so that breath, your respiration, converted oxygen into carbon dioxide. And so respiration consumes oxygen, lowering oxygen concentrations in the water. Whereas gross primary production produces oxygen. So it's increasing the oxygen concentration in the water. And importantly, production occurs only during the daytime. So we can track changes in dissolved oxygen from day to night using dissolved oxygen sensors. And then we can kind of relatively easily estimate those metabolism metrics of respiration and GPP. And we can do this in both streams and lakes. And in general, stream metabolism is more closely linked to nutrient load, whereas lake metabolism is more closely linked to trophic state. And remember that trophic state is a slow variable. And lake metabolism metrics are closely tied to those water quality differences that we've talked about between oligotrophic and eutrophic lakes. So we have these eutrophic lakes with these really high phytoplankton, kind of murky water, and they generally have very high rates of GPP and R. They have high nutrient concentrations, a lot of biological activity. In contrast, those oligotrophic lakes, low nutrient concentrations, really low rates of GPP and R. And kind of as an oligotrophic lake transitions to a eutrophic lake, it will exhibit increasing rates of GPP and R. And importantly, we can detect those changes in GPP and R before you can detect changes in nutrient concentrations and phytoplankton or algae concentrations. So it's kind of this early indicator of change, really sensitive metric. So lake metabolism can be used kind of as this ecosystem vital sign, where it's both the indicator of current trophic state, and if monitored through time, it can be used to detect changes before other more commonly measured variables. Okay, so lake metabolism is generally measured in the middle of the lake in these open water areas, but those kind of single point measurements don't really indicate how these different streams might be contributing to that eutrophication process. And we know from that long-term study that mitigating stream nutrient load is a really urgent kind of concern in this lake, and that stream nutrient load contributes to that eutrophication. So streams are causing changes in this lake. And so far, I've just been discussing kind of stream versus lake metabolism, but we could actually think of freshwater metabolism along a spatial continuum from streams to lakes, where metabolism responses in streams are more dominated by fast variables, and then metabolism in lakes are more dominated by slow variables. And in this study, we want to see if metabolism in the lake near that stream-lake transition zone could be directly related to inflow stream drivers. So people have looked at kind of spatial patterns in lake metabolism, but they generally kind of compare open water to nearshore areas. And here we want to strategically target that stream-lake transitional zone. And so because open water metabolism is this well-established, really sensitive measure of change, if we can see a response within that stream-lake transitional zone, it might provide an even earlier indication of that kind of impending ecosystem change. So kind of an early warning of that early warning. So in this study, we estimate lake metabolism in nearshore areas to where streams inflow to the lake. So this is a map of our sampling locations. We have one open water site, which served as a reference for where metabolism is most commonly estimated, and then two nearshore sites, one located near a warm stream inflow. The water in the stream is generally the same temperature or even warmer than the surface of the lake. So it likely mixes directly with the nearshore surface water. We would expect if this stream is contributing to eutrophication, it would be stimulating higher GPP and R at this site, and that those higher rates of GPP and R would be at least partially due to those stream-related drivers. And then remember that these higher rates of GPP and R signify that worsening kind of water quality condition. So we also looked at a site near a cold stream inflow, and this stream, the water is generally cooler and therefore more dense than the surface water of the lake. So that stream tends to drop more towards the bottom of the lake, having less mixing with that surface water where we're taking that metabolism measurement. So if this stream is kind of disconnected from that site's metabolism, we would expect to see no relationship between the stream-related drivers and the GPP and R response. At each site, we deployed oxygen sensors hanging off of buoys, recording dissolved oxygen every 10 minutes during the summer of 2018. We then used this process-based model to convert the dissolved oxygen measurements into the metabolism metrics of GPP and R. And we identified higher rates of GPP at both nearshore sites in comparison to the open water site, with a significant difference here with unique letters above each of the box plots. And then kind of note that we have this on a log scale y-axis. You can see that the higher rates of R at the warm site in comparison to the open water site, but there's no significant difference between the cold stream R and the other sites. So this kind of generally follows the previously identified patterns where we see higher GPP and R in nearshore areas, but we want to figure out if any of that kind of higher magnitude of R could be attributed specifically to the nearby stream drivers. So we conducted a stacked ensemble machine learning driver analysis to identify what proportion of that GPP and R response at each site could be attributed to those different driver variable categories. We looked at lake and meteorological drivers, which are kind of common in these lake metabolism studies. And then we also included stream-related drivers. So things like stream depth, stream water versus the lake water kind of as a mixing potential metric, for example. And overall, we found that 20% of the elevated GPP at the warm stream site could be attributed to stream-related drivers, whereas at the cold stream site, that machine learning analysis did not detect a large portion of that site's GPP. Did not detect that a large portion of that site's GPP could be attributed to our driver variables. So for R, we found that over 10% of the response at both sites could be attributed to those stream-related drivers, though that warm stream site was also kind of highly dependent on meteorological drivers. So this provides evidence that both of the streams can affect nearshore metabolism, and that warm stream site, which has a much greater kind of stream-lake mixing, at least surface mixing potential, has the highest rates of GPP and R. So it indicates that that nearshore metabolism is likely stimulated by that load, the nutrient load in the stream. And overall, by looking at that stream-lake transitional zone, we can, and including those stream-related drivers, we may not only get this kind of early warning of change even sooner than that classic kind of open water metabolism estimates, but we could also get this more detailed information about how each individual stream might be contributing kind of differentially to that eutrophication process, which would help managers kind of target specific areas within that watershed. Okay, so kind of in summary here, we use this metabolism model to kind of translate those DO measurements into metabolism, enabling us to kind of better understand those cross ecosystem connections that may be driving the lake ecosystem status. And of course, this has management implications to incorporate kind of strategically located sampling to indicate what areas of the watershed might be driving changes in the lake. And I think what's important for thinking about that kind of spatial targeting of areas in the watershed is that it's much less time intensive to get these metabolism estimates than doing a really detailed nutrient load analysis of all of the inflow streams. So has kind of a lot of management savings implications. Okay, so we have these temporal and spatial studies that have this assessment of the conditions in the lake. And then we want to think about specific things we could do as leverage points for change. So some sort of management intervention to better achieve the water quality goals. And so to do this, I met with the director, the executive and the research director at the LSPA to kind of brainstorm solutions. And the majority of the land in this watershed is private property. So working with private property owners is absolutely essential. And because the water quality and the overall condition of the lake is so high, the executive director, June, highlighted that it can be really hard to communicate this kind of sense of urgency that's needed to maintain this oligotrophic lake into the future. And she wanted some sort of intervention that could kind of light a fire or get people motivated and excited to act. So we decided to test the use of a simple model visualization that would connect today's land management decisions with future water quality. And this visualization would directly connect the fast variables of daily decision making and the slow variables of long-term ecosystem change. In this study, we ask, can interactive data visualizations promote waterfront best management practices? And here we focused on how people perceive the connections between ecosystem services and their individual actions. And really, this visualization works to kind of close this time delay between actions and the resulting changes to ecosystem services. And in lieu of observing kind of individual actions, we ask people about their behavioral intentions or what they plan to do on their property in the next year. Okay, so we focused on two best management practices. One, installing a waterfront buffer, so having natural vegetation between the home site and the water, and then avoiding lawn fertilizer application. We mailed invitations to lakefront property owners in the watershed, and then participants could log on to their personal computers. They would complete an online questionnaire, and at the end of which they would automatically be directed to use either an interactive or a non-interactive visualization, and they were kind of randomly assigned to these groups. And then they completed a follow-up questionnaire. And let's see, so we had these two different visualizations. The non-interactive was just kind of this regular website with information on how buffers and fertilizers relate to water quality outcomes. It had this kind of view from the dock photo to communicate that key ecosystem service of Lake Sunapee, which is water clarity. And users would scroll from the top to the bottom of the page, and as they went, they'd see more examples of different types of ways in which buffers and fertilizers affect water quality. In contrast, the interactive version had drop-down menus on the left where users would select specific property management decisions, and we had the exact same type of visualization on the right side of the page as the non-interactive website. And so this kind of controlled for visualization style. The only difference is this level of interactivity. So users can select property management decisions, and then they see different kind of outcomes, update, select other properties, see those changes in the visualization. So we found that participants became more likely to adopt best management practices after using either visualization. Here are the results for the fertilizer application. So this is a distribution density plot, similar to a box plot. And the thicker portions kind of indicate where most of the points fall, and then those thinner portions indicate where fewer points fall in the distribution. And then I added this darker shading to kind of highlight that middle range of the middle 50% of participants. Okay. So we generally see this median shift greater than 0 for both of them. And since it's 4:43, I'm just gonna skip ahead a little bit. We see this increase, and we see this kind of similar pattern for waterfront buffers. So most people are becoming more likely to install the buffer after using the visualization. Getting to the interesting stuff is that we found that seasonal residents were more likely than permanent residents to shift their fertilizer intentions. So this watershed is a really popular kind of vacation home area. And so you have this interesting, maybe tension is not the right word, but an interesting dynamic between the folks who live year round in this area versus the folks who have their kind of second home summer seasonal resident. So we see this difference between where seasonal residents are much more likely to avoid that fertilizer application after using the visualization. And we think this could be related to other research that has found that seasonal and permanent residents may kind of care more about different types of ecosystem services. And since our visualization focused on one specific type of ecosystem service, mainly water quality, it could make sense that people that are more drawn to that area specifically because of that water clarity might be more convinced by a visualization that focuses on that ecosystem service. They also may have been less exposed to other kind of environmental education outreach within this particular area because they're only living there seasonally. So kind of overall, we see that using this simple model visualization can connect those kind of today's decisions with the future water quality, and could help identify specific methods for the LSPA to reach out to kind of specific groups within the watershed. Okay, so I'm going to skim past, in the interest of time, just summarizing, reiterating the things that I've already said on this. And then super quickly talk about what I'm starting to get into with my new role at the Minnesota DNR. So I've been in this role for about 3 months. I still have a lot to learn, working on kind of getting settled and learning what our Upper Mississippi River Restoration Program does. This is a 5-state, 3-agency partnership, 3 federal agency partnerships. So there are 6 field stations kind of spread across the Upper Mississippi River Basin. And I'm the supervisor for that Lake City station, which is in Minnesota, and the kind of furthest upstream station within the program. We have over 30 years of monitoring data and a programmatic kind of focus on linking science and management. And I'm particularly interested in kind of building capacity within our field stations, specifically through academic partnerships, and where I think we could kind of leverage the strengths and needs of both our program and having these access to this 30 years of monitoring data, and kind of building on the strengths of academic institutions, perhaps graduate student training, et cetera. So one thing that I'm starting to look at kind of in this large interdisciplinary and cross-sector group is how the Resist, Accept, Direct, or the RAD climate change management framework may help transform Mississippi River management. And this RAD framework focuses on these action words of resist, accept, and direct. And that kind of explicitly recognizes that management is this ongoing, active process. It's not something that you just do and then you're done. And this framework also explicitly focuses on examining and managing ecosystem variability and those kind of dynamic processes of change. And I think it has a lot of promise in kind of helping managers and researchers integrate those dynamic processes of large rivers and working across those interjurisdictional boundaries, considering those kind of cross-scale interactions in this large river system. So that's just one brief highlight of what I've gotten into in the first 3 months, but a lot more to come, and excited to kind of be digging back into my roots here in the Upper Midwest. Overall, I hope that this research kind of highlights how strategically selecting key fast variables that initiate change in those slow variables could maybe further our understanding of these complex dynamics in human natural systems. And also how integrated science and management, when scientists meet stakeholders where they're at, with kind of the full recognition that scientific knowledge is only one piece of that complex human natural system, that we may kind of better understand the diverse ways in which people are connected to water, so that we can manage our precious freshwater ecosystems for the next generation. So with that, thank you, and would love to discuss or take questions.